Compound containing heme. Many porphyrin containing metalloprotein have heme as their prosthetic group, that is, heme and the proteins they are joined together. For example, myoglobin, methemalbumin, methemoglobin, sulphemoglobin, cytochromes, catalase, HaemPeroxidase, endothelial nitric oxide synthase. These are the groups which contain, or the compounds that contain heme combined with the protein globin. Myoglobin. Myoglobin is a single heme-polypeptide complex. It is present in muscles and binds oxygen with its heme group like hemoglobin. Myoglobin is a monomeric protein that has 154 amino acids residue. When I'll go to the next slide to the diagram, I'll explain it to you that what you mean by the monomeric protein and how it consists of 8 alpha helix connected through the turn with an oxygen binding site. It has a globular structure. Here you can see monomeric, that is a single protein chain is there, or single monomer is there, one heme, one protein chain. Whereas the hemoglobin molecule is a tetramer. You can see 2 alpha chains, 2 beta chains, 4 protein chains and 4 heme molecules are there. So it is a tetramer, whereas the myoglobin is a monomer. And the other point in the last slide was that 8 alpha helix are there. You can see here there is no broken chain. There is single polypeptide chain and the helix are there. 8 helix molecule, 1, 2, 3, 4, 5, 6, 7, 8. 8 helix, and these are joined together. Therefore, we say that a single chain. Within a single chain, the spring-shaped coils are there, and therefore we call it that it is a monomer. It is a single large polypeptide chain folded together in 8 alpha helix shape. Primary structure. Primary structure of myoglobin, hemoglobin. It contains, what do you mean by the primary structure? Primary structure means that the long chain showing the amount of the amino acid. 4 things are mentioned in the primary structure. That is the number. You should know the number of amino acid, the amount of each amino acid, the sequence of the amino acid, and the polypeptide chain is open or closed. That is, either it is straight chain or it is closed. Then we come to the secondary structure. Secondary structure means that it is a folded structure. The protein takes a shape of a helix, that is a coil structure, or it is present in a sheet form, which is known as the beta sheet. Then the further folding of the secondary structure takes place and it forms a tertiary structure. Tertiary structure means that either it is a fiber-like, as it is found as the protein keratin, which is found in here, fiber-like, or it is a globular shape. Globule, you just imagine the globule of a boiling egg. If we put the egg in boiling water, then it will take the shape of the globule. Quaternary structure. Quaternary structure means that arrangement of the monomer in such a way to make a complete functional unit. Right? I'll go back to the diagram. Suppose that if these chains are separated, if these monomers are separated, they will not function. Right? If I keep these apart from each other, they will not function. But the arrangement in this way, that is the arrangement of the tetramer, is functional. And in this, Just follow my pointer. There are the hydrogen bonds in between the monomers, and these monomers are connected together by the hydrogen bond, and in this way the tetramer, it becomes a complete functional unit. Myoglobin contains a heme prosthetic group which is responsible for carrying oxygen molecule to the muscles. Myoglobin exists in the oxygen-free form, deoxymyoglobin, just like hemoglobin, oxygen-free form and oxygen-combined form. Plasma concentration of myoglobin may rise if skeletal or the myocardial cells are damaged. In this case, the myoglobin is released in the plasma. Because of its smaller size, it's rapidly excreted by the kidneys, and increased amount in the filtrate may cause the renal damage. Because if it is filtered through the glomerulus, then it may cause the renal damage. Methemalbumin, an abnormal compound formed in the blood as a result of heme combining with plasma albumin. It gives brown color to plasma and occur in hemolytic and hemorrhagic disorders. Methemalbuminaemia is a clinical condition that can be caused by severe intravascular hemolysis, acute hemorrhagic pancreatitis due to the proteolytic breakdown of the hemoglobin. Bleeding into the abdominal cavity is another cause of methemalbuminaemia. Methemoglobin is an oxidized derivative of hemoglobin that has iron in the ferric form. You see, in the ferric form. Therefore, it cannot bind or carry oxygen, because the ferrous iron can only bind or release oxygen. The ferric state cannot bind or release oxygen. It is brown and normally present in low plasma concentration. But there is an enzyme, NADH-dependent enzyme, Methemoglobin reductase. It is responsible for converting methemoglobin into the hemoglobin. So this enzyme, it can convert. Methemoglobin can be converted to hemoglobin. For the one correct answer you can note down. So the methemoglobin can be converted to the hemoglobin by the NADH-dependent enzyme Methemoglobin reductase. Symptoms of Methemalbuminaemia: hypoxia, which causes cyanosis. Cyanosis is defined as a bluish coloration of the skin, and it is due to the poor circulation, poor oxygen containing capacity, inadequate oxygenation. So cyanosis is due to the inadequate oxygenation, and basically due to the inadequate amount of the hemoglobin, and it can also occur as increased respiratory rate. Sulphemoglobin, sulfur, is a green pigmented molecule which is not normally present in the body. It is made by the oxidation of iron in hemoglobin to the ferric state, and which is so it is formed by the oxidation of iron from ferrous state to ferric state. And who is responsible for this oxidation? The drugs and the chemicals that contain sulfur are responsible for the oxidation of iron from ferrous to ferric state. Sulfur binds to hemoglobin molecule which forms sulphemoglobin. It cannot be reconverted to hemoglobin. Note it down. Sulphemoglobin cannot be converted into hemoglobin. Cytochromes. Cytochromes are protein containing heme as a cofactor, and they are classified according to the type of heme and its mode of binding. The iron in cytochrome, it exists in the +2 state and a ferric state and ferroxo state. It depends upon the oxidation and reduction. That is, when the electrons are moved from one form to another form, then iron can be oxidized or it can be reduced. Cytochromes, just now I have told you that cytochromes are capable of performing electron transfer due to reduction or oxidation of their mean iron. The location of cytochrome depends on their function. They can be found as globular protein and membrane protein. In the process of oxidative phosphorylation, globular cytochrome C protein is involved in the electron transfer. You have seen the process of oxidative phosphorylation, and cytochrome, they are responsible for the electron transfer and help in the transfer of the electron from complex 3 to complex 4. Catalase, a very common enzyme, a well-known enzyme, and it is found nearly in all living organisms exposed to oxygen, such as bacteria, plants, and animals, and it catalyzes the decomposition of hydrogen peroxide to water and oxygen. It is a very important enzyme in protecting the cell from oxidative damage. Because, why? Because it decomposes hydrogen peroxide to oxygen and water. One catalase molecule can convert millions of hydrogen peroxide molecules to water and oxygen easily. Catalase is a tetramer composed of 4 polypeptide chains, each over 500 amino acids long. It contains 4 iron-containing heme groups that allow the enzyme to react with the hydrogen peroxide. And you can see here hydrogen peroxide is decomposed to water and oxygen. HaemPeroxidase. As the name indicates, heme-containing enzymes that use hydrogen peroxide as electron acceptor, and then the hydrogen peroxide is decomposed. This is a two-electron oxidation reduction reaction. Hydrogen peroxide is reduced to water and iron in the enzyme is oxidized. We'll just see the reaction and then this statement will become more clear to you. One oxidizing equivalent resides on the iron given an oxyferrite state. Here we can see iron is present in the ferric state. It decomposes, the hydrogen peroxide is decomposed into water, and the iron, it is converted into the ferrite state. In this way, one electron is transferred to the water, as well the one it is resides on the iron. Now, when this electron is lost, in this way, when this compound loses the electron, then this electron is lost and this is then converted to the compound 2. This compound 2 is then again converted. It loses an electron and it is again converted to ferric compound. Endothelial nitrogen oxide synthase 3 is an enzyme that in humans is located in the chromosome 7. This enzyme is one of the 3 isoforms that synthesize nitric oxide. Nitric oxide, a small gaseous and lipophilic molecule that participate in several biological processes. Nitric oxide is primarily responsible for the generation of nitric oxide in the vascular endothelium. Vascular endothelium, what is vascular endothelium? It is a layer of flat cells lining the internal surface of the blood vessels. At the interface between circulating blood in the lumen and remainder of the vessel, just imagine it's just like a bag which lines the blood vessels, and the blood is flowing within this bag inside the blood vessels. Or you can imagine it as a pipe, a very delicate pipe inside the blood vessels, which is made up of monolayer of flat cells, and blood is flowing within this pipe inside the blood vessels. Functions of nitric oxide produced by endothelial nitric oxide synthase. It is very essential for a healthy cardiovascular system, since nitric oxide produced by endothelial nitric oxide synthase in the vascular endothelium plays a very important role in providing the vascular tone, cellular proliferation, leukocyte addition, and the platelet aggregation.